Low-power diagrams are usually drawn to show the distribution and proportion of different tissues in a specimen. For example, the low-power diagram on the right shows the distribution of vascular tissues in the transverse section of a part of a dicotyledonous stem. Let's learn how to draw a low-power biological diagram of the dicot stem shown in this photomicrograph. Using a sharp HB pencil, draw the outlines of the tissues with single and continuous lines. Do not draw any individual cells. Draw freehand. Do not use a ruler or a pair of compasses. Make sure that the different parts are in proportion. The drawing should resemble the specimen. Do not copy diagrams from books. Do not shade the structures. Label the relevant structures. Put labels on the sides, using straight labeling lines. Note that the lines must not cross one another. Lastly, write a title below the diagram. This should include the name of the specimen and the power of magnification. If you are drawing a cut surface of a specimen, you have to state in the title how the section is cut, such as transverse section, T, S, or longitudinal section, L, S.